Sweden, and the title is Immunostimulating Gene Therapy. And I will talk to you about preclinical results, but also from our clinical trials that are ongoing in Uppsala. And these are my disclosures. So what is immunostimulatory gene therapy? Well, it is the transfer of immunostimulatory genes into the tumor milieu, aiming to induce an anti-tumor immunity. And if you look in the tumor microenvironment, a lot of things are actually going on that is not really positive for immunity. So the tumor itself can produce a lot of cytokines and chemokines that will attract immature myeloid cells from the bone marrow to the tumor, and these are very suppressive. They also convert macrophages to an M2 profile, which in turn also inhibit immune responses. And then myeloid-derived suppressor cells and other players in the tumors, they promote the development of T regulatory cells in the tumor. So these players together produce a lot of immunoinhibitory cytokines like IL-10 and also growth factors like TGF-beta, IL-35 and so on. And together this will inhibit both the activation of dendritic cells but also the activation of cytotoxic T cells and NK cells. So if we don't have any active antigen-presenting cells such as dendritic cells, we will have no activation of de novo T cell responses. And for those T cells that are already there, they will be blocked from entering the tumor. For example, macrophages can somehow be guards that prevent the T cells to enter. As I said before, we have no M1 activation of the macrophages since they are shifted to M2. And we have the T cell inhibition. So how can we sort of move further here? How can we make the tumor microenvironment become more positive for immune responses? So there are a lot of things you can do. And what will in the end activate T cells are dendritic cells. And they are activated through a lot of different molecules. But one of the most potent stimulators of dendritic cells is CD40 ligand. But you can also affect the T cells directly with gene therapy, for example, by trying to provide co-stimulatory molecules via the tumor cells. And here you can look what the dendritic cells have. Well, they have the B7 family, or as I call them here, the CD80 and CD86. There are a lot of different cytokines and chemokines that you could insert into the tumor area, so you will have an influx of tumor-specific T cells. So I have been working with adenoviral vectors for quite some time, and first we have worked with an adenovirus serotype 5 and its replication incompetent through an E1, E3 deletion. And we have tried a lot of different gene inserts, and I will show you some of the data from those. We started working with bladder cancer, urinary bladder cancer, and the aim here is then to make the virus with the different transgenes come into the tumor and make the tumor produce immunostimulatory molecules. So the tumor cells themselves can activate dendritic cells that in turn activates T cells or NK cells to kill the tumor. So this is an example of a survival curve treating urinary bladder tumors in mice with different viruses, the CD40 ligand and IL-12 and also the B7 family. And in these studies, we saw that CD40 ligand gave a really good survival in these mice, and especially we noted that the mice that were cured from their tumors, they were also immune if you tried to inject the tumor again. So we went further to combine compare CD40 ligand to other molecules like trans and also lymphotactin, which is a chemokine, and also IL-2 and IL-15. And in this experiment, together with a set of other experiments, we still thought that CD40 ligand was the most potent molecule. So we went further to analyze what really happened in the tumors that were treated with CD40 ligand. So So we decided to take biopsies when we couldn't really see a difference yet with a control virus and the CD40 ligand virus. So we took biopsies here, and then we did a lot of different analyses. And we could see that in the tumor we had an increase of IL-12, for example, which is a very stimulatory molecule. And we had a decrease of IL-10 and TGF-beta in these tumors, and those, are suppressive, so it was good news for us that they were decreased. In another set of experiments, we also looked at the M1 macrophages, and we could see that when you treat animals with CD40 ligand, you will have a shift from the M2 macrophages to M1, and that is also really good, since the M1 macrophages can not only help with good cytokines for the T cells, but they also can kill the tumor cells directly. We also noted in these tumors that myeloid-derived suppressor cells were decreased. We do not know if it's because the tumor doesn't attract them anymore, or if they are depleted or maybe converted to another cell type when we have them together with CD40 ligand. That is still under investigation. When we looked at the T cell infiltration, we could see that tumors that are treated with CD40 ligand, they have a large infiltration of T cells, both of CD4 T helper type and also the cytotoxic T cells. And these cells, if you take out T cells from the mice, you could show in vitro that these T cells can kill the MB49 cells, which are the bladder cells in this case, but they don't kill irrelevant cells. So you really gain T cells that are specific to the tumor. So one thing that we looked further in why the T cells are infiltrating into the tumor, it can of course be that a lot of different cytokines or chemokines are released that attract the T cells, but we know that the endothelial cells are actually CD40 positive, and if you stimulate endothelial cells with CD40 ligand, they should be able to upregulate molecules that are important for T cell attachment, rolling, and transmigration. So we analyzed human endothelial cells that were stimulated with our viruses, and we could see that molecules that are very important for this process were increased when they were stimulated with CD40 ligand. So I think that this is one of the components that make the T cells go into the tumor. Now, what I presented so far is mostly, of course, murine work, and you need to know before you start clinical trials if it's also relevant in the human system. And we tried to stimulate human dendritic cells with CD40 ligand virus, and we could see in a series of experiments that they get highly activated and produce a lot of cytokines, for example, IL-12. And they produced IL-12 even in the presence of IL-10, which is otherwise a suppressive molecule that is also present then in the tumor. So far we concluded then that CD40 ligand can really kickstart the immune activation in the tumors by activating dendritic cells that in turn activate the T cells and so on. We also know that the CD40 ligand induces an M1 shift from M2 macrophages. They reduce suppressor cells of different types. We can promote T cell entry into the tumor, and of course then induce tumor cell apoptosis. So with this in mind, we started a clinical trial, and this trial was done for bladder carcinoma. Bladder carcinoma is actually an ideal tumor to start gene therapy because the bladder cavity is fairly isolated from the rest of the body, so you could inject the viruses in the bladder without them spreading in the whole body. And we started a trial in 2006, and well, the scientific community were still worried a little bit about gene therapy, so we thought bladder cancer is fairly safe. It's also easy to access, to take biopsies and so on. It's non-invasive since you can go through the ureter. Normally, when the tumors are small, like in in the picture here, you can do a transurethral resection. So you just go in through the urethra and take out the small tumor. And then you add, for example, BCG, which is an immunotherapy by itself, or you add chemo in the bladder. And this usually prolongs the time before the tumor comes back. But then you have tumors that are starting to be invasive, so they are going a little bit infiltrating into the muscle layer around the bladder. And this is actually a very good time point to try novel therapies. Because this patient has a couple of weeks of time before they can actually do the surgery, from the time of when they know that they need to do a surgery to remove the whole bladder. If you don't remove the whole bladder, these patients will not be able to be treated anymore. So during the time they waited for surgery to take out their bladders, we were allowed to give them 3 installations of virus directly into the bladders. And we treated 8 patients. And after the treatments, the bladders were taken out, and we could do, well, a lot of interesting investigations, and you could actually have access to the whole tumor. So it was a unique and interesting situation. Also, these patients, they don't have large amount of tumors. It's rather low dose of tumor, you could say, in this patient. And that also means that their immune system is fairly well. So we investigated these patients, and I will show you examples from 3 of them on histochemistry. And before treatment, you can see that these 3 patients had tumors in the biopsies, so it were indeed tumor patients. And when we treated them, the first patient, you could see really massive infiltration of lymphocytes close to the urothelial lining. We couldn't find any tumor cells, no high-grade tumor cells anywhere in that bladder. And patient number 2 was the same thing. We saw inflammation, but no tumor. And then the third patient didn't have a response at all, did not have any inflammatory cells in the bladder, and we could still see the tumor. So when we moved further with the analysis, we wanted to know if the T cells were active, because I mean they can just be there but become energetic. But we saw increased interferon gamma in these bladders, and systemically we could also see a reduction of T regulatory cells. And the overall trial conclusions was that CD40 ligand gene therapy was well tolerated. We didn't see any adverse events in these patients. They didn't have fever even, and you expect that since it's an immunotherapy, but maybe it was the sort of localized treatment in the bladder that made the difference in this case. We could also see then the T cell infiltration, and they were active since they produced interferon gamma. And in the end, 4 of the 8 treated patients had no remaining tumor in the bladder. One of them we could see a tumor higher up in the urethra, but otherwise no tumor. The final 3 patients that we treated, we moved on to treat these earlier patients that you could have the small tumors that you could just take away, but our treatment didn't really have any effect on those, and I think they look too normal. You need to have tumors that look really ugly for the immune system to be able to see them properly. So about this time point, we came in contact with Henrik von Euler, who is a veterinarian in Uppsala working at the animal hospital, and he wanted to use our virus to treat dogs with melanoma in his clinic. And we got permits to do that, and here is one example of a dog that has malignant melanoma in the gum. And we were giving this tumor 1 injection, and after this, almost the whole tumor was gone. This is another example when we have a tumor in the eyelid, so it's an uveal malignant melanoma. And we treated this tumor 6 times with CD40 ligand virus, and we could see that the tumor started to shrink, and we also saw a large inflammation in the eye, They started to lose some hair close to the eye, and after that the tumor was completely gone, and after a few weeks also the hair came back. So all in all we treated 19 dogs with melanoma, and we saw 5 complete responses. We had 8 partial responses, 4 stable disease, and only 2 didn't respond at all and had progressive disease. So we thought these data really supported to do a trial also on patients with malignant melanoma. So we did that, and this is then really late stage patients that were even resistant to ipilimumab, which is another immunotherapy. So, but you have to start there at least. So we treated the first cohort of patients, which were 6 patients with CD40 ligand virus. And then we continued with another 9 patients, and then we added cyclophosphamide before the first and 4th treatment. And we do this as a preconditioning and also supportive chemo to try to suppress a little bit the T regulatory cells to increase the chances for immunotherapy to work, since you usually do that also for other immunotherapies like T cells and so on. So this time we were giving the treatment by percutaneous intratumoral injection. Most patients that we treated, 15 all in data I will present today, they most of them had liver metastasis. So this is an example of patient number 7 who had a liver metastasis and were treated with 4 injections. And you could see that after treatment we have a significant reduction of the activity in the tumor. The same was seen in patient number 12, also a large metastasis in the liver, and in this case the tumor activity was almost lost. We did a survival curve, and we could see that cyclophosphamide seemed to increase the survival at 6 months, but I mean these are few patients, so you have to interpret it to be a little bit cautious when you interpreted this data. In the end, 2 patients in each group survived more than 2.5 years, so in the end we didn't see a difference between these 2 groups. We looked at some immunology also in these patients, and what we could see was that the patients that responded to treatment, and these are shown here in red, they usually had low levels of T regulatory cells, or they became at least low after cyclophosphamide, and they also had higher levels of activated T helper cells and activated cytotoxic T lymphocytes. And these are then analysis in the blood, so we couldn't do that on tumor samples. So trial conclusions here is that if you do PET-CT images, we saw an effect in 7 out of 15 patients. But in the end, we are measuring MRI pictures using the RECIST criteria, and then the best responses were still stable disease. And then we had stable disease in 6 out of 15 patients, and 2 of them survived more than 2.5 years. So where will we go from here then? There are a lot of things that you can do along the line, and I think that the CD40 ligand virus really have potential. But we got this virus from Baylor College of Medicine, and they didn't take any patents, and we didn't take any patents for one of the diseases, like a use patent. And when you don't do that, nobody will move further to take your virus and make it a commercial available product. So that's how it is. So I got in contact with a Swedish billionaire, and he told me that we should pimp CD40 ligand virus and make a new one. And I did that, and we call this load 703. And this virus has an improved capacity compared to the CD40 ligand virus because, first of all, it's oncolytic. And for me, to have it oncolytic means that it will at least spread the virus a little bit more than if it was replication incompetent. It also means that we will have maybe a little bit longer expression of the transgenes since more cells will be infected. And we also will have some spreading of antigens. We also changed the fiber on the virus to that of serotype 35, so we will target instead CD46. And CD46 is more abundantly expressed on tumor cells compared to CAR, which is the normal receptor for adenovirus serotype 5. We also made it double armed, and that means that we didn't only have CD40 ligand in this virus, but we also added 4-1BB ligand. And CD40 ligand now, have a lot of interesting effects that would be good to use in the tumor microenvironment. But 4-1BB ligand will actually potentiate dendritic cell stimulation, and it will also act directly on the T cells and NK cells that are already present in the tumor to promote their survival, expansion, and also memory development. And we did a set of experiments to show that the virus was better. And here, load 700 is the CD40 ligand alone in a similar otherwise construct, and load 703 is then our new virus. So we could see that the dendritic cells stimulated with load 703 had a much higher level of co-stimulatory molecules, and we could also see that the level of IL-12 and IL-21 was really enhanced. If you take these activated dendritic cells and use them to stimulate PBMCs from CMV-specific donors, you can actually see that we will have a great expansion of generally of T cells in these co-cultures. We will also have a stimulation of antigen-specific T cells, like the CMV-specific T cells in this case. But most of all, we see an enormous expansion also of NK cells in these cultures. And of course, we did studies on a series of different cell lines, but focused on pancreatic cancer cell lines to see that the virus really was oncolytic and it killed most of the cells in culture. We also saw this capacity, oncolytic capacity in vivo in animal models. We are still working on the immunostimulatory aspects in animals, which is quite tricky since these viruses doesn't infect urine cells very well when you change it to the 35 fiber. So in summary, CD40 ligand is a prominent activator of anti-tumor immunity. We know that CD40 ligand therapy is feasible and seems safe. We have some effect in patients also. And we know that 4-1BB ligand can potentiate the effect of CD40 ligand, and that makes load 703 a very interesting candidate for immunostimulatory gene therapy. And we are currently setting up a clinical trial for pancreatic cancer, and that we will do at Baylor College of Medicine in the US. So there have been a lot of people involved in all of this work, especially when you do clinical trial. It really escalates with collaborators, which is fun. And I would like to thank them all. And if you have any questions, I'm happy to take them now. Yeah.
I'm interested in your print version. So on its own, you couldn't get a pattern on the single gene.
No.
But in a bicistronic form, you could.
Yes, well, we actually made also some different, a different version of CD40 ligand, you can say, and I can't disclose exactly how it looks right now.
Well, that's my question. Why couldn't you just do CD40 ligand?
That is already protected by another company that is working. No, well, the modified I can make a patent of, and that I'm doing right now. But I can't use wild type CD40 ligand. Yeah, that one is protected. Yeah. So if there are no more questions, oh well, we have one final question.
Thanks, very fascinating talk. I wanted to ask a little bit about the arming still. Have you compared this virus with GM-CSF coding virus? Because that is sort of becoming the first approval
we haven't compared it to a virus that has GM-CSF, but we have done some studies where we added GM-CSF to urine tumor cells. And it seems to be, I mean, GM-CSF has so many different roles, and if you have the wrong concentration, you will have an influx of bad cells. You will have mildly derived suppressor cells instead, or you could even hamper the activation of disease, and that we could see. But if you have the right concentration, on the other hand, you may promote that you get infiltration of dendritic cells, for example. So we haven't compared them directly head to head, but I think actually that it would be interesting to have both in the same vector, because if you have influx of disease, CD40 ligand will make sure that they will be good cells and not bad.
Can I ask one more thing?
Yeah.
So then you mentioned in preclinical data that you can infect the epithelial cells with a non-replicating virus, right? And they start expressing CD40 ligand, and then there's trafficking, better trafficking, right? So how can the epithelial cells then produce the CD40 ligand if it's E1 deleted virus? Because they are not mutated cancer cells.
Yeah, well, we have done different experiments here, and the first I showed you here with the HUVEC, the human cells, there we used our old CD40 ligand, so it's an Ad5 replication deficient virus with a CMV promoter, so they are easily transduced with the Ad5 virus. We have also used our oncolytic virus, and they are a little bit sensitive, even if they shouldn't, but when you culture them like this, they might have some a disruption in the RB pathway.
Okay, yeah.
So yeah.
Thanks.
Thank you. Then this session is ended here, and the conference will actually begin now within, was it 4? Or 5? We are a bit early because Alexandra is not here. So thank you all for listening.